The next point now I want to turn to is just how numerous microbes are. So let's ask a very simple question. How many microbes are there on Earth? And to bring this into a human reference point, let's begin with looking at the numbers of the human population. So I'm from Los Angeles, which at the latest census was around 10 million people. And in the state of California, we're up to approximately 35 million. And in the United States in general, nearly 300 million. These are large numbers, but overall in the world, we're up 3 orders of magnitude at 6 billion people. And that's a lot of folks. However, this is nothing in comparison to the microbial population, as estimated by a wonderful paper that I'm citing here at the bottom of this slide called Prokaryotes, the Unseen Majority, that was published in PNAS in 1998. And these are very rough numbers, but give or take an order of magnitude here or there, I think you're going to be impressed when you see the number that I'm about to show you. So the estimates for the microbial population are just enormous, 5 times 10 to the 30th cells. And this indeed is such a large number that it's very difficult to wrap our minds around it. So to try to make this a bit easier to do, I did a very simple calculation where I assumed that the length of a given microorganism was 1 micron and asked, how many times would we need to go back and forth between the Earth and the Sun if we lined up all of these organisms end to end in order to account for this number? And the answer, shockingly, is we would need to go back and forth 200 trillion times. So hopefully that impresses you with just how many of these creatures there are on the planet. Now, where are they? If there's so many, how come we don't think about this all the time? Why aren't we overwhelmed? Well, one reason is that oftentimes we're shockingly ignorant about the fact that they're all around us, that we ourselves are really walking microorganisms. So one of the first scientists to appreciate this profound fact was the father of microscopy, Anthony van Leeuwenhoek. And this is a lovely image that he drew from his observations down his first microscope in 1684. And you can see he drew some nice rods and cocci, and even pictures of probably motility, what's meant by these dotted lines from C to D. And he reflected as he was looking through the microscope about his own teeth. And this is, I think, a very funny quote. He said, "Though my teeth are kept usually very clean, nevertheless, when I view them in a magnifying glass, I find growing between them wet matter as thick as a wetted flower. The number of these animals in the scurf of a man's teeth are so many that I believe they exceed the number of men in a kingdom." Well, this indeed is actually an underestimate. Not only do they exceed the number of men and women in a kingdom, they go far beyond that. And so if we actually look at our own bodies, just take a look at your wrist and 1 square inch on the surface of your wrist. Right there, we're estimated to have 5 to 50,000 bacterial cells. And it just increases in density as we move to other parts of the body, such as the groin and the underarms, in our teeth, and really where it's mainly at in our bodies is in our colon. And the overall total per person is 70 trillion. That's quite a lot. And one thing that I think is really important for you to know about the microbial community within your own body is that there are 10 times the number of microbial cells in our system than there are human cells. And not only that, but when we look at the genetic potential of the DNA within these organisms, the genetic potential of only those within our guts is over 100 times that of the human genome. So you might begin to ask whether or not humans are not merely walking vats of microorganisms, carriers serving their existence. It's something to think about, and there's a great deal of research now emerging that's beginning to illuminate just how crucial these organisms are for human health, not only with regard to being able to help us digest our food, but also interfacing and controlling our immune system in ways that are fascinating and profound. Now, despite the fact that this number, 10 to the 12th, seems really large, and indeed it is, it's peanuts when we compare to other domains that we find microorganisms. So let's start where they're least abundant, up in the air. It's quite amazing to me that they have been detected as high as 34 to 46 miles up into the sky. But these concentrations are really small relative to other compartments. As I told you, within the human body, we have quite a few, and when you add up all of the humans and domestic animals, and then termites, which I'll get back to in just a bit, the order of magnitude jumps up to about 10 to the 23rd to 24th. This is superseded by the quantities that you can find in soils, in forests or grasslands, desert, tundra, and swamp environments. These places are very fertile homes for microorganisms, and their activities can transform the chemistry of their environment quite profoundly. And this is, of course, also true in aquatic domains, where at similar orders of magnitude, we find microorganisms in both marine and freshwater environments. But all of these numbers pale in comparison to the numbers that we find in the subsurface, both in terrestrial and oceanic environments, where microorganisms have been detected as deep as 2 miles. Now, this really is a very interesting frontier area in microbiology. It's hard to go down into these depths, and yet nowadays researchers are being equipped with the tools they need in order to access these remote communities. And what remains to be learned is what exactly these organisms are doing in situ. Are they active? And if so, what are their activities? Are these activities affecting in a significant way the physical and chemical properties of these environments? We don't know, and we look forward in the coming decades to finding the answers to these and other interesting questions. So now let me just give you an example, a tour through various parts of the world and other inhabitants of that world where we find these organisms, just to bring home to you how ubiquitous microbes are on the planet. So to start with what might be a more familiar image, here what you're looking at is pond scum. You're looking at a wonderful assemblage of phototrophs and other microorganisms in this pond. And my favorites, of course, are these purple phototrophs. These are the ones that I told you about earlier that are what we call the anoxygenic phototrophs, that are not utilizing water as a substrate in photosynthesis, but are utilizing other more reduced compounds, such as different types of sulfur species, hydrogen, or iron. Now, these organisms that we see in modern-day ponds, as I told you at the beginning when I was illustrating the antiquity of microbial life with the example about banded iron formations, are absolutely historically important for their metabolism and the diversity of their metabolism and how it's changed the geochemistry of the Earth. Not only has the evolution of photosynthesis contributed to evolving our atmosphere to one that contains oxygen over the course of time, but as I also showed you with the banded iron formations, these type of organisms have likely shaped ore formation as well. And many other important processes have been able to come about thanks to these organisms doing what they do. And it should be noted that this type of metabolic activity, photosynthesis, is one that today we're highly interested in because of our need for coming up with alternative energy sources. And certainly, if chemists were able to mimic what these wonderful microbes in this pond do, we would be able to not worry so much about our dependence upon foreign oil and our fossil fuel supplies being burned. But that's a story for another day. The point is, their metabolic diversity is old, you see it all around us, and the biochemistry is really quite fascinating. Continuing on with the chemistry and the metabolism of these organisms, not only do they do important things when they're growing, but they also do important things when they start hitting what we call stationary phase. And this is a point in their development where they're not necessarily actively growing, but they're at a higher density and they're just hanging out metabolically. And when this occurs in their life cycle, sometimes metabolites and pigments begin to be excreted. And these pigments, which are called secondary metabolites, although that name itself may be a bit misleading because they're only secondary in a temporal sense, in that they're made after a phase of active growth. But by no means are they secondary in terms of the physiology of the organisms that produce them. Nonetheless, these metabolites oftentimes are used today by pharmaceutical companies as natural products that confer antibiotic activity. And a terrific example of this are organisms in the streptomycete family that you see here in this petri dish that are producing a whole host of wonderful antibiotic compounds. Now, containing in the environment of the soil, of course, are roots of plants. And in this part of the soil, what's known as the rhizosphere, we can find microorganisms as well that are colonizing in a very beneficial way the plant roots. And here is a tomato root seedling. This is an image taken by Guido Bloomberg, and he showed in experiments in the laboratory when he took tomato root seedlings and mixed them with an organism called Pseudomonas, that this bacterium was able to colonize the plant and form what we call biofilms on the surface of the root. And this is just one example of organisms that interact with plants. There are many that fall into this category with different names. And the bottom line is that they have a very beneficial relationship with these plants, where sometimes they produce natural products that fend the plant off from fungal predators, and so they serve as biocontrol agents. Other times, these organisms are capable of fixing molecular nitrogen into a usable form and essentially acting as a natural fertilizer. Now, crawling around in not only soil environments, but of course we're very familiar with these from our homes, are termites. And the termites are a terrific source of microbial diversity and one that's becoming an increasingly important microenvironment in which to look because of our desire to understand microbial processes that might be harnessed for lignocellulose degradation, again out of a need to develop alternative sources of energy. Now, a colleague of mine, Professor Jared Ledbetter at Caltech, studies these termites, and he likes to call them an ecosystem in a microliter. And I think this is really a fantastic description of them because it's within their hindgut that you find a zoo of microorganisms and protozoa that are swimming around doing all sorts of important activities that make it possible for the termites to digest their wood. And in the process, they emit methane. And not an insignificant fraction of this methane ultimately makes its way up into the atmosphere and contributes to the overall chemistry on the planet. So speaking of methanogens, here you see a dramatic illustration of them at work. This image that I'm standing in front of is taken from Cedar Swamp in Woods Hole, Massachusetts. And it's an image of a group of students from the Microbial Diversity class, which is a fantastic course for about 20 students, half from the United States and half from overseas, who come together every summer to understand how microorganisms are able to perform these various metabolic activities that I've been describing in these lectures. And what you can see here is that the students have gone waist deep into this swamp, and they've stomped around, and as they've done this, they've collected the bubbles that come up as they stomp the sediment and collected them in these inverted funnels. And then some brave individual holds that funnel and removes their hand just at the moment when a friend comes by with a flame and ignites it. And here you see a lovely illustration of methane at work. So methanogenesis led to the creation of the methane gas that was ignited here. Now, in the past, the activity of these organisms that generate this methane, that are called methanogens, might have been important in shaping the chemistry of the Earth's environment. And the reason we suspect this may be the case is because early in Earth history, the environment contained appreciably more methane than it does today. Now, a different example of a habitat where microorganisms are very important is in Chile and in other places on Earth, but this example here is taken from the Andina copper mine in the Andes in Chile, where microorganisms are exploited for their abilities to help with bioleaching. And so what happens is that in these mines, there are piles that are built up, and they're fertilized essentially with indigenous microbial populations that are able to live in shockingly low pH levels, down to pH as low as 1 and sometimes even lower. And these organisms are essentially eating the minerals in this mine pile, and in the process of metabolizing it, changing the mineralogy in such a way that copper is solubilized and leached. So here's another example of an environment that is quite extraordinary, and yet microorganisms have been able to adapt and even to thrive in this extreme condition. So on our tour of extreme pHs, we just saw an example of low pH. Let's now go to a high pH environment. This one I'm showing you is Mono Lake, that is in Northern California. And Mono Lake is quite an extraordinary place. It looks almost like it's from another planet. You see these beautiful tufa towers that are calcium carbonate minerals forming, and it's because the pH is so high and the alkalinity is so high that they naturally precipitate from these waters. In addition to having these carbonate minerals contained within this lake environment, is a ton of arsenic, and I'll get to this in part 2 of my lecture today. And what I want to point out right now is that in this very high pH environment, and also one that's replete with arsenic, nevertheless we find organisms called alkalophiles that thrive here, that are able to make a living utilizing arsenic as a terminal electron acceptor in respiration. This is the subject of my second lecture. And in so doing, account for 14% of the carbon turnover in the system. Now let's go on to another example of an extreme environment. Here now we're looking at an extreme of salt. And there's no better example of this than the Dead Sea in Israel. But you can find organisms such as those that inhabit the Dead Sea also in the Great Salt Lake and other places on Earth, such as salt flats, where you have very high salt content. And the organisms living here are capable of growing despite this high salt and have adapted particular molecular strategies to cope with it. One very elegant example of this is their ability to use special photopigments called rhodopsins, and these are colored purple. And these rhodopsins they have in their membranes and enable them to generate energy under conditions where they need to use slightly different strategies than organisms that are growing under conditions that we would consider more normal. Now, so approaching the end of our tour through microbial diversity and ubiquity, I want to end with a few other extremes now that are based on temperature and pressure. If we think about the extremes of cold, there's no better place to go than Antarctica. And you might be surprised to realize that even in this environment, you have microorganisms thriving in the crust. And these organisms are called psychrophiles. And their ability to grow is dependent upon dust from winds carrying nutrients picked up from the continents surrounding Antarctica, South America, Australia, Africa, that reach Antarctica, deposit their dust, and fertilize these upper crusts of the ice, where we have intrepid pioneer organisms that are able to utilize these nutrients and grow, even in these very cold regimes. So another extreme is that of temperature and pressure. And there's no better environment in which to observe this than at the bottom of the ocean, in environments where we have hydrothermal vents that are releasing nutrients into the deep. And here's an example of one of these vents. It's called a black smoker because the nutrients that it releases, including manganese and iron, often precipitate in the conditions of the oceans at these sites such that they look black. Now, around these vents, there's abundant life, really extraordinary life, not just microbial life, but giant tube worms and fish and other macroscopic organisms. Okay, so the ability of all of this abundant life to be in this environment crucially depends upon the activities of microorganisms that are chemosynthetic, that are able to grow by the oxidation of sulfur and other compounds that you have present in this environment, and couple that oxidation of these reduced substrates to the fixation of CO2 into biomass. And this is at the base of the food chain that then sustains the growth of other marine organisms, such as these tube worms. And here you see an example of that. And these beautiful tube worms, if you cut them open and you look at one of their organs called the trophosome, within these organs are bacterial symbionts that are doing the process that I just mentioned. So my final example that I'll end with is one that might be the most familiar to you if you've ever done any PCR in molecular biology. So most of you have heard of the enzyme Taq polymerase. And this polymerase is what allows us to do an amplification reaction when we're doing PCR. Now, this enzyme Taq derives from a bacterium called Thermus aquaticus. That's where the Taq comes from. The T is from the thermus and the ac from aquaticus. And this is a thermophile that was isolated in Yellowstone at a hot spring many decades ago, and it was presciently realized by Kary Mullis and others that the enzymes contained within it could be useful for various biotechnological applications because they wouldn't denature at the temperatures that would kill most other types of cells. So these thermophiles are a very fascinating group of organisms whose molecular adaptations include not only DNA polymerases, but also a wide variety of other enzymes that might be of industrial use.